reason plants are green is because our chlorophyll molecules will be absorbing blue and red light, but then reflecting green light back. That's what we see. Yeah. So number two, what is the difference between chlorophyll and chloroplast? Yeah, good. So the chlorophyll, left one up on the screen for you, are the pigment molecules that are absorbing the light, and then they are actually located inside of the chloroplast in the thylakoid membrane. And just like Clara said, the chloroplasts are the organelle where all of these processes take place. Any questions about these? All right, so a couple of things I neglected to mention. This series of reactions, so the absorbing of light energy and the splitting of water to produce oxygen, are called the light reactions. And so that's because they are, of course, using light. And so these light reactions are going to be taking place in the thylakoid membrane. So we'll put that on our diagram right here. So they're going to be pumping protons from the stroma here into the inside of the thylakoid membrane, and then using those protons to create a buildup of both ATP and then NADPH, which are going to accumulate in the stroma. So those are going to be important for our next step in photosynthesis, which is called the Calvin cycle. So the Calvin cycle is going to be taking place in the stroma. Here. And so part of the reason that it needs to take place in the stroma is because it is dependent upon the light reactions for the ATP and NADPH. All right. What's that?
It's dependent upon light reactions.
For ATP and NADPH. Yeah. So if we think about what we just talked about with the light reactions and look at our equation for photosynthesis, we can see that we've accounted for some of the components thus far. So we've used water, we've used light, and we've produced oxygen. But what we haven't done yet is we haven't used CO2 and we haven't produced any glucose. And so that's what we're going to be doing with the Calvin cycle. Yes?
What exactly does oxygen play? What is oxygen created in this process?
So oxygen is created from the splitting of water. So if you remember, we strip an electron from the chlorophyll to send it down the electron transport chain, and then we need to replace that electron. So we do that by splitting the water to produce the oxygen with protons.
Right.
All right. So the Calvin cycle can be presented in a pretty complicated way. Much like Rick was emphasizing last week with the reactions of glycolysis. If you go on to upper level biochemistry class, you'll probably have to learn the Calvin cycle in really painful detail, but I'm not going to do that to you. So the Calvin cycle, basically what we're doing is we are taking carbon dioxide and then we're fixing it into sugars. So that term fixing is what we talk about when we take carbon from an inorganic molecule like carbon dioxide and put it into an organic molecule such as sugars. And so this is a cycle. So we're going to start out with a 5-carbon molecule. This is RUBP. And then we'll start off with 3 of those. And then we're going to add 1 carbon dioxide to each of those 5 carbon molecules. So we've got 3 RUBPs, so we'll need 3 CO2s. So the enzyme that's catalyzing this reaction is called rubisco. Here's a 3-dimensional rendering of rubisco. So it's got an enormous name, which is actually pretty informative. So ribulose 1,5-bisphosphate carboxylase oxygenase. It's a mouthful. You don't need to remember that. So the ribulose 1,5-bisphosphate is this guy. So it's our 5-carbon sugar molecule. And the enzyme rubisco can either carboxylate or oxygenate that molecule. So we'll start off by talking about the carboxylation. So all that means is just adding the CO2 to the molecule. And so after rubisco does its thing, we're going to wind up with a 6-carbon molecule. So let's say 3 6-carbon molecules like that. But as it turns out, those 6-carbon molecules are actually really unstable. And so they pretty much immediately break into 3-carbon sugars. So we had 3 6-carbon sugars. We're going to split all of those in half. So that'll give us 6. Diane on me here. 6 3-carbon sugars. And so from there, we're going to reduce our 3-carbon sugars. So we're going to use the energy from ATP and then also the electron from the NADPH. So those are the 2 components that we created with our light reactions. And this is where we're going to use them. So we'll go through a couple of steps. Now you don't need to worry about remembering the numbers of ATP and NADPH that we're using here. You just need to know that for each cycle that we go through, we're going to be incorporating 1 carbon dioxide. So each time we go through the cycle, we capture 1 carbon from the atmosphere.